Hello, everyone, and welcome to this GEN Biotechnology webinar entitled Tales of Innovation, Enhancing RNA Therapeutics via Novel Poly-A Modifications. As the potential of mRNA-based medicines expands, researchers are making exciting strides in overcoming the challenges associated with the intrinsic instability and immunostimulatory properties of exogenous mRNA. By introducing various modifications across the entire mRNA sequence from cap to tail, translational activity can be enhanced and immunogenicity risks reduced. This webinar will discuss novel modifications to the poly-A tail to improve the stability and translational activity of RNA therapeutics. This webinar was made possible through support from TriLink BioTechnologies. I'll be the moderator for this event. My name is Fei Lin, Senior Technology Editor at GEN. We're pleased to welcome our speaker for today's event, Dr. Chunping Xu, who is the Senior Director of Chemistry R&D at TriLink BioTechnologies, where she oversees the development of novel clean cap analogs for improved protein expression and process optimization for modified NTPs. Leveraging expertise in nucleotide chemistry, Dr. Xu and her team have created a library of over 100 novel cap analogs to identify next-generation clean cap analogs. Before joining TriLink, she had extensive experience in small molecule drug development and conjugation. Make sure to stay tuned for our live Q&A at the end of today's presentation. If you have a question, please submit your questions in the Q&A box.
Thanks for the introduction, and also thanks everyone for joining this webinar. Today, I'm going to talk about tales of innovation, enhancing RNA therapeutics with novel poly-A modifications. So the first part of this webinar, I'm going to give you some background about what is poly-A tail, what's the function, and why do we want to modify the poly-A tail, and what are the methods to attach a tail modification. The second part of this webinar, I'm going to introduce TriLink's new poly-A tail modification technology and talk about its impact on mRNA protein expression and longevity. The last part of this webinar, I will touch briefly about how to optimize your mRNA design from cap to tail. So first, why do we want to modify a poly-A tail on mRNA? Before that, a little bit of background about mRNA. As most of you are familiar with mRNA, mRNA is a messenger between DNA and the protein. It takes genetic information from DNA and from the nucleus and uses it to translate into protein in the cytoplasm. In recent years, especially after the pandemic, mRNA start to find a wider application in different areas. The most well-known example is using mRNA as a COVID vaccine. mRNA can also be used in gene editing, cell therapies, personalized medicine, protein replacement, and non-translated RNA. So the question is, why do we want to introduce chemical modification to mRNA? Historically, mRNA has been known to be very unstable, and it can trigger immune response. Also, as a large molecule, mRNA is very hard to deliver to the cells. Good news is technologies have been developed to overcome these mRNA challenges. Chemical modifications have been introduced to mRNA to stabilize mRNA and also reduce immune response. And also new delivery methods such as LNP and other delivery methods have been developed to overcome the delivery challenges. So here is the sequence of mRNA. Chemical modification can be introduced from the cap to the base and to the mRNA tail to optimize your mRNA performance. So in today's webinar, I will touch briefly on cap modification and base modification. I will mainly focus on mRNA tail modification. So TriLink has been in the mRNA field for more than 10 years. We are the leader in the co-transcriptional mRNA capping technology. Over the years, we have developed various generations of CleanCap analog. And we also own IP for all those CleanCap analogs. So CleanCap M6 is our newest cap analog, also the best in class. Compared to other capping technologies such as ARCA and enzymatic capping, CleanCap M6 can significantly increase your mRNA protein translation. Base modification has also been explored a lot. The pioneer research was done by Nobel Prize winner Katie Carrico and Drew Weissman. When N1-methylpseudo-U is introduced into EGFP, it can significantly increase protein translation at the different cell lines. Other base modifications such as pseudo-U, 5-methyl-C, 5-methoxy-U also have similar effects. So compared to cap and base modification, poly-A tail modification has not been explored a lot. So before we go into poly-A tail modification, first, let's try to understand what is the role or function of the poly-A tail. The first function is the poly-A tail protects mRNA from 3' to 5' degradation by exonuclease. And also the poly-A tail binds to PABP protein and further binds to eIF4E and eIF4G protein complex. Together with the cap, it modulates the mRNA translation. So what are the potential benefits of modifying the poly-A tail? First, by modifying the poly-A tail, we can stabilize the mRNA and make it more resistant to exonuclease cleavage. And it can increase the mRNA longevity and duration. Also, by stabilizing mRNA, we can potentially improve the mRNA translation efficiency. So now the question is, what are the approaches to attach a modification to the poly-A tail? Here are the common methods we can use to introduce tail modification. We can use RNA polymerase, such as poly-A or poly-U polymerase. mRNA tail modification can also be introduced through RNA ligation and also chemical conjugation. And also, we can introduce tail modifications through DNA template and IVT reaction. So here are some examples of published work about mRNA tail modification. Some of you probably have read the work from Dr. Xiaowang's lab at MIT. So they used the RNA ligase to attach a modified oligo strand to the mRNA tail. By doing that, it can significantly improve the mRNA protein translation. Later on, they published another paper and showed that using a branched and modified tail, it works even better. Another paper is published by a research team from Hong Kong Technology University. They introduced tail modifications through DNA template. By introducing around 20% C to the poly-A tail, it can significantly increase mRNA duration and also protein translation. Now I'm going to introduce TriLink's poly-A tail modification, and I'll show you the impact on protein translation and also mRNA longevity. So this is the process flow we used to screen various different tail modifications. So starting from a purified mRNA, first we attach poly-A tail modification. After purification, the modified mRNA is subject to cell-based assay for protein translation, and the promising candidates is moved to in vivo mice study. So using this process flow and also use EGFP as a model, we have screened various different tail modification in two different cell lines, 293T and A549. Compared to EGFP without tail mod, all the tail modification were able to increase the overall protein translation in the two different cell lines. We picked the mod 22 to launch as poly A modification service for two reasons. One, mod 22 is one of the best. And two is we have generated a comprehensive data package for mod 22. So in order to apply the tail modification to the mRNA manufacturing process, we need to address some questions our customers may have, since every customer is working with their unique mRNA sequence. The first question is, does the tail modification work for various mRNA constructs with different size? And also the second question is tail modification compatible with mRNA with different poly A tail lengths? And also, we would like to show, is tail modification compatible with mRNA with different caps and base modifications? Lastly, we also like to explore whether tail modification changes mRNA immunogenicity. So we test our tail modification in 4 different mRNA constructs with different size, from EGFP to F-loop to beta-gal to Cas9. All this mRNA has 128 tail lengths. After tail modification and purification, we were able to get good HPLC purity and good tail modification for all the 4 mRNAs. Next, we also test our tail modification with mCherry with 88 tail lengths. So this left table shows here is a mRNA quality comparison between the 2 mRNA pre and post tail modifications. This data shows that the 2 mRNA has comparable capping efficiency, purity, double-strand RNA level, and also the tail modification was done with very high integration efficiency. On the right graph, it shows the cell assay data for protein expression for mCherry with or without tail modification. The top orange line is mCherry with tail mod, and the bottom blue one is mCherry without tail mod. And we can clearly see at the later time points, mCherry with tail mod have significant increase in protein translation. And we also see a shift of peak protein expression from the blue to the orange to a later time point. The next question we want to address is our tail modification compatible with mRNA with different tail lengths? So we made EGFP with 40A, 60A, 100A, and also 120A tail lengths, with or without tail modification. All the mRNAs were subject to cell-based assay. And the top orange is EGFP with tail mod, and the bottom blue is EGFP without tail mod. And this results shows us, regardless of the tail length, our tail modification works nicely to increase protein translation and also mRNA duration. So far, we have shown that the tail modification is compatible with different mRNA construct with different tail lengths. Now the question is it compatible with different cap and a different base? So EGFP is made with or without tail modification, and with 2 different caps, CleanCap AG3' methyl and CleanCap M6. And also the EGFP is made with N1-methylpseudoU base modification. From these cell assay results, we can clearly see that regardless of the cap analog, our tail modification work very nicely to improve protein translation for both caps at later time points. And also we can see a clear and nice shift of peak protein expression from unmodified EGFP to tail-modified EGFP. So far, all the cell-based assays have shown that our tail modification works successfully to improve protein translation and mRNA duration. And it's compatible with various mRNA with different tail lengths, different caps, and base modification. Next, we also would like to check whether we can see the same impact in vivo mice study. And also we would like to check the mRNA immunogenicity with the tail modification. Here is the exciting in vivo mice data using F-Look with CleanCap AG3' or methyl. So F-Look with or without tail mod will formulate with LNP and inject into mice. And this left graph shows the mean protein expression from 7 mice up to 7 days. And the top orange line is F-Look with tail modification. And the bottom blue line is F-Look without tail modification. And we can clearly see the significant increase in protein translation and also mRNA duration. And the middle graph is the beautiful luminescent image of the in vivo mice study. The top group of mice is control mice with PBS. And the middle group of mice is with F-Look without tail mod. And the bottom mice is F-Look plus tail mod. And at early time points, 12 hours, we see slightly increase in protein translation with tail mod. And the later time point at 48 and 72 hours, we start to see dramatic difference between the F-Look with or without tail mod. The F-Look with tail modification show much longer duration of protein expression. And the right graph is the actual quantification of protein translation. And we can see that at 72 hours, compared to the mice with F-Look without tail mod at the baseline, the mice with F-Look plus tail mod is still expressing decent amount of protein. We also did the in vivo mice study with our best cap, CleanCap M6. And I would like you to focus on the middle graph directly for luminescent image. At 12-hour time point, since we are using a better cap now, both the F-Look with or without tail modification showed a high level of protein expression. But at longer time point, 48 hours and 72 hours, we start to see clear difference between the F-Look with tail modification and without tail modification. And again, we confirm the longer mRNA duration in this in vivo study. This is great news for mRNA therapeutic application. Higher protein expression, longer mRNA duration means we can use low dose of mRNA or less frequent mRNA dosage. That also means less immunogenicity and less side effects. Other than the reported F-Look, we also tested mRNA in vivo mice study with a different mRNA construct, human EPO. So this left graph shows the mean protein expression from 7 different mice up to 4 days. The top orange line is EPO with tail modification, and the bottom blue line is EPO without tail modification. And we can see at the longer time point, especially at 72 hours and 96 hours, we saw a significant increase in protein translation and mRNA duration. And the right bar graph shows the overall protein expression. Compared to EPO without tail modification, EPO plus tail modification has 1.25-fold increase in total protein expression from 4 days. Next, we also would like to see that EPO with tail modification, whether it can trigger any immune response. So we looked into 2 inflammatory cytokine and 2 chemokine expression in the mouse serum at different time points, 1 hour, 6 hour, and 24 hour. The orange bar is EPO mRNA with tail modification, and the gray bar is EPO mRNA without tail modification. And we didn't see significant difference between the EPO with tail modification and without tail modification for all the cytokines and chemokines we have checked. And also compared to the positive control, which is mRNA with Y-type U and ARCA. Both of the EPO mRNA has very low immune response. Again, this is very good. This is great news for tail modification. We don't want to introduce any extra immunogenicity. So to summarize our tail modification technology, here are a few key takeaways. First, this tail modification is a very easy to plug in process for current TriLink's mRNA manufacturing process, and it can be done with very high efficiency. And also, our tail modification is compatible with different mRNA construct, different base modification, different caps, and different poly-A lengths. Most importantly, our poly-A tail modification can significantly increase your mRNA protein translation and duration. So we recently launched this poly-A modification service in the TIDES conference. Please check our website for more details about how we offer this service. So the last part of this webinar, I would like to touch briefly about mRNA design, how to optimize your mRNA design from cap to tail. So using all the technologies in hand, we should introduce the best capping, best base modification, and best poly-A tail modification to the entire mRNA sequence. Together with 5' UTR and 3' UTR sequence optimization, and also codon optimization on the open reading frame, it can maximize your mRNA performance. To support your mRNA innovation, TriLink has recently developed various technologies, new products, and service. So as a recent TIDES, TriLink has launched all these new services and new technologies. First, we start to offer mRNA sequence optimization offered by our Ofsina Bio team. They recently got integrated into TriLink. Also, we are launching CleanCap IVT kits for higher yield, almost double as regular IVT yield, and a low double-strand RNA. And we also launched the custom mRNA with poly-A tail modification service. Other than that, we also launched the custom and catalog self-amplifying RNA service, and also plate-level library synthesis of guide RNA. So the last slides, just a little bit about who we are. TriLink BioTechnology is based in San Diego, California. So we are not only a cap analog company, we also offer mRNA service. So we offer IUO and GMP mRNA synthesis, modified NTP synthesis, and also mRNA cap. We also offer custom oligo synthesis and also guide RNA synthesis. We have a decent size R&D team. We are doing a lot of mRNA innovation, and we are looking forward to collaborate with you. With that, I'd like to thank you for attending this webinar. I'm happy to take any questions you may have. Thanks.
That's a wrap for today's webinar. Make sure to stay tuned for our live Q&A. If you have a question, please submit your questions in the Q&A box. Hello, everyone. Thank you, Dr. Xu, for that great presentation. And we will now begin our Q&A session. And again, if you have a question, please submit them in the Q&A box. So our first question says, how do you verify the incorporation of the tail modification?
Good question. We normally use the HPLC method to detect, to monitor the tail modification efficiency. So on HPLC, we can see different retention time for modified mRNA and unmodified mRNA.
The next question says, does the tail modification change the mRNA poly A tail length?
It doesn't. So it still maintains the same poly A tail length, but we just add a small chemical moiety to the end of the poly A tail.
Have you tried the tail modification on saRNA?
Very good question. We are still investigating it. Preliminary results shows it also works on saRNA, but more to update soon.
Does the poly A modification only impact the tail or through the ORF?
It doesn't change your ORF. It only changes the tail.
Great. The next question asks to elaborate a bit more on the details of the modification.
Due to IP status, we couldn't disclose too much about what is the tail modification, but our PCT will publish in July, so you can find out more after that.
Next, audience member says, great talk. Curious to know how many modified A's are essential to see the effect.
Could you repeat that question?
Yes, how many modified, I believe poly A's are essential to see the effect?
We have screened a lot of like 30 to 50 different tail modification. So on different tail lengths. So far we, for most of the tail modification we have tested so far, we saw the effect.
Great. All right, the next one says, how was the modification introduced into the poly A tail?
So it's through one of the tail modification approaches I described in one of the slides. But again, details I cannot reveal now. But you can find out later on once our IP is secured.
Does the modification occur only at the end of the poly A tail, or is it introduced within the internal adenosines as well?
It happened only at the end of the poly A tail.
Is there a further loss in mRNA after purification?
Actually, after the tail modification and purification, the mRNA recovery is pretty high, normally around 90%.
What is the mechanism of action of the modification? Does it affect poly A tail binding?
Very good question. The detailed mechanism we are still investigating, and we had some preliminary results using a model study that shows by using our tail modification, it can slow down the exonucleus cleavage at the 3' end of the poly tail. And okay, sorry. And the second question, does it change the PABP binding? We still investigating it.
Did you compare expression of mRNA with poly A tail against post-IVT poly A with poly A polymerase?
I I need more clarification for this question. So I can describe what we have done so far. So we compare mRNA without tail modification and with tail modification, and we see, significant impact from the tail modification. And we haven't directly compared with, poly A polymerase modification, the other tail modification methodologies yet.
Right, we have a few more questions. What is the modification's effect on mRNA encapsulation?
We have tried. It doesn't. The general answer is it doesn't impact the mRNA encapsulation because we have tried different lipids and different kind of LNP formulation, and it all worked same way as the regular mRNA.
Does this modification impact the closed loop structure?
We believe it doesn't, because if impact the closed loop structure, then it will interfere with the protein translation. But again, we need to investigate more about that part.
Great. Is there a limit on poly A length or an optimal length for the modification?
So far, we have explored from 48 to 128, and we believe this tail modification should be independent from tail length. It should work for whatever your tail length. But yeah, what we have tried is from 48 to 128. And the optimal should be 80 to 128.
Are you incorporating different poly A modifications in the same mRNA or the same repeated modification several times?
We incorporating just one modification, one simple modification to the poly A tail. So we are not combining different modification into one mRNA.
Great. All right, looks like that might be the end of our flood of questions. Plenty of comments in the chat that says great talk. Thank you so much, Dr. Xu, for that fantastic deep dive into RNA modifications. And thank you to the audience for your questions and joining us for this GEN Biotechnology webinar. Thank you to TriLink for their support. So this webinar will be made available on demand within 48 hours, and we hope to see you next time.
Thanks, Fei, and thanks for all the audience.